Agrotech students, and we are here to do an experiment about Auxin. Auxin is a plant hormone that causes cell elongation, and it is also responsible in regulating plant growth. Auxin is produced in the cells of the apical meristem and is transported to various portions of tissues and cells around the apical meristem, signaling these cells to elongate. Areas with less concentration of auxin have growth temporarily stunted.
So for example, this mulberry tree is still growing and it has a lot of auxin. And when it reaches its final stages of its life, definitely its auxin concentration will be less.
The function of auxin is auxin influence rooting formation. Auxin can also promote flowering by specifying the site of flower initiation. Auxin can also be used as a weed killer.
Now we are going to start the experiment by getting 8 stems of mulberry stems, and each stem has 4 internodes. So internodes are this part of the plant of the mulberry trees. So I'm gonna cut slantedly so that the auxin can be absorbed more efficiently. So it's slantedly cut. Now I will divide the trees with 8 parts into 8 stems. So as we can see here, these are the internodes. For each stem, we need to have 4 internodes each. So this one has 1, 2, 3, 4. So we cut here slantedly. And then repeat the same thing. 1, 2, 3, 4. Two pots that is divided into, it is labeled as the first pot is auxin and second pot is no auxin. So with these pots there are 4 stems each. Okay, so with the auxin, we need to dip in the water first, and here is the auxin. So firstly, we need to dip inside the water, the slanted part. Second thing is the precaution is the stem need to be pointing upwards. So I did a mistake here. This one is pointing downwards, so it's actually supposed to be like this. So firstly we need to dip inside the water. Then dip inside the auxin. Then we need to tap off the excess. Like so, and then inside the soil. So I need to repeat the process for the same thing. Okay, now for the no auxin part, we just dip inside the water and with all the stems pointing upwards. We already have planted this, so we need to wait for 3 weeks for it to grow and see the results after 3 weeks. The results of this experiment is obtained after 3 weeks all of the mulberry stem growth. As we can see here, the stems are removed one by one from the pot. Next, we are removing the mulberry stems that are applied with Auxin one by one. These are the stems that have been applied with Auxin. As we can see, there are a lot of leaves that can be counted, and they are dark green in color, which means they are healthy. And for the roots are going down with gravity, and for the stems without Auxin is lesser in comparison. Here are the results for the stem that are applied with Auxin. As we can see, there are clearly root growth and the leaf growth. This is because Auxin promotes cell elongation. And here is the picture for the stems that are not applied with Auxin. We can clearly see the difference, which are there are lesser leaves or no leaves at all, and there are no roots at all or only one strand of root.